Bismillahirrahmanirrahim, my dear first year student and BDS class. Now we continue to the next organelle, that is the mitochondria. You know mitochondria very well in the previous classes, in FSC classes, and in the ordinary lectures from the biochemistry and physiology. The mitochondria is a very important and very well-known organelle present in the cytoplasm. It is double membrane organelle. It is the powerhouse or the grid of the cell. Greek word mean mitos mean thread and chondrial mean granule. It is a thread-like granule, mean it is smaller in size. Greek word, it is double membrane bound organelle found in all eukaryotic cell. Mitochondria are commonly present between, commonly sized is about 0.75 and 3 millimicron in diameter. Usually specifically stained, they are not visible. तो ये बात ध्यान में रखें कि ऑर्डिनरी विजिबिलिटी ऑर्डिनरी स्टेन से नहीं होती. In EM it is also visible, but with special stain it can be made it to be visible. Number of mitochondria vary in cell to cell. Those organs, those tissues which require more energy to perform their function, they will present more in number. For example, in red blood cell, see there is no mitochondria, but in the liver cell and muscle cell is the rich mitochondria present because of their utility at that tissues. No, अब एग्जाम्पल के तौर पर हम अगर देखें तो आयन ट्रांसपोर्टिंग सेल जो है वो एक बेस्ट एग्जाम्पल है. Iron transporting cell, you all know these are the transporting cells, especially seen in the kidneys and anywhere in the duct system. You see iron transporting cell, and iron transporting cell having a basal infolding. Basal infolding, you see in this diagram, it is the electron microscopic picture. Basal infolding means that the basal plasma lemma, they invaginate within the cell to form a double layer inside the cytoplasm, forming a folding pattern. It is called basal infolding. So this basal infolding is to increase the surface area. Basal infolding means to increase the surface area. So you see there are a lot of mitochondria present within the wedges of these basal infolding. इनके अंदर जो है mitochondria numerously present, because over here there is the utility and also the site location, because यहाँ पे ही mitochondria को होना चाहिए, because always there is a constant turnover of the various solvents, solutes, these are the micronutrients, also the iron, they are transported from basal side. So from where there is this mechanism energy dependent as well, because the active transport is constantly carried on in this basal infolding. So this is the situation of mitochondria in the basal infolding. So जहाँ पे इसकी ज़रूरत ज़्यादा होगी माइटोकॉन्ड्रिया की, वहीं पे जो है इसके नंबर भी बढ़ेगा और इसके बाद इसकी एक्टिविटी भी वहाँ प्रोमिनेंट होगी. और यही वजह है कि माइटोकॉन्ड्रिया जब स्टेन लेता है eosinophilic, so that is why the pinkish area become prominent in these iron transporting cell. ये भी याद रखें हिस्टोलॉजिकली, जहाँ माइटोकॉन्ड्रिया ज़्यादा होंगे वहाँ पे स्टेनिंग जो है eosinophilic होगी. So basal eosinophilia is the characteristic feature of iron transporting cell. क्या फंक्शन परफॉर्म करता है? सप्लाई cellular energy for the aerobic respiration, quite prominent and quite known and very well known function of the mitochondria, provide provision of energies. Then the signaling the calcium and phospholipid metabolism में ये help करता है. कैल्शियम चैनलिंग में भी कैल्शियम रेगुलेट करता है. इसके बाद लिपिड मेटाबॉलिज्म में भी और फॉस्फेट मेटाबॉलिज्म में भी इसकी रोल है. सेलुलर डिफरेंशिएशन एंड सेल डेथ, ये भी याद रखें कि सेल की जब एक सेल बाय बडिंग डिवाइड होता है या 1 to 2 to ordinary mitosis, so there is the division of the mitochondria as well. So our mitochondria on both the daughter cells will be equally distributed, and there is this mitochondria is then role of the cell differentiation and also cell death. सेल डेथ मीन एप्रोप्टोसिस, अगर वो पोराम है या नेक्रोसिस है। एप्रोप्टोसिस इज द पोराम सेल डेथ, अगर पोराम सेल डेथ है उसे हम कहेंगे एप्रोप्टोसिस। वैसे जो है जो सेल इंसल्ट हैं ड्यू टू एनी काज, उससे जो डेथ होती है उसे नेक्रोसिस कहते हैं। सेल डेथ नेक्रोसिस एंड एप्रोप्टोसिस में भी फंक्शन जो है वो परफॉर्म करता है mitochondria, because the mitochondria is the first target organelle for the degeneration and necrosis, also as the apoptotic cell. Control of the cell cycle, obviously cell cycle जो है वो control करता है from during resting phase, during replicating phase, it's also provide a important function. Cell growth, mitochondria biogenesis in turn temporarily coordinated with these cellular process. तो सेल ग्रोथ में माइटोकॉन्ड्रियल बायोजेनेसिस भी प्ले रोल करता है सेल की कोऑर्डिनेशन के लिए। माइटोकॉन्ड्रिया हैज़ बीन इम्प्लिकेटेड इन सेवरल ह्यूमन डिजीज लाइक कार्डियक डिसफंक्शन, हार्ट फेलियर एंड ऑटिज्म। तो ये कुछ डिजीज ऐसी हैं जिनमें अगर माइटोकॉन्ड्रिया फंक्शन प्रॉपरली नहीं कर पा रहा, सो देयर आर मैनी अदर डिजीज विच शुड बी विजिबल इन द नॉर्मल ह्यूमन, दैट इज द कार्डियक डिसफंक्शन। कार्डियक जो कोऑर्डिनेशन एक्टिविटी है उसमें भी कैल्शियम आयन का रोल है और माइटोकॉन्ड्रिया का भी डेफिनेटली रोल है। हार्ट फेलियर एंड ऑटिज्म और द थ्री अदर देयर आर मैनी अदर एग्जांपल, बट व्यू ओवर हियर हमने सिर्फ 3 की बात की है, कार्डियक डिसफंक्शन, हार्ट फेलियर एंड ऑटिज्म। Now we come to the histological structure. The outer mitochondrial membrane consists of the outer mitochondrial membrane, the inner mitochondrial membrane, the intermembranous space, that is the space between the outer and inner membrane, the cristae formed by the infolding of inner membrane, the matrix space between the inner membrane and the mitochondria with stripped outer membrane are called the mytoplast. तो माइटोप्लास्ट की क्या डेफिनेशन है कि हम अगर स्ट्रिप कर दें आउटर माइटोकॉन्ड्रियल मेम्ब्रेन को, देन द माइटोकॉन्ड्रिया इज कॉल्ड एज माइटोप्लास्ट। Now, this one is the beautiful picture of a electron microscopic of the mitochondrion. Mitochondrion आपको यहाँ 2 नज़र आ रहे हैं. This is the outer membrane, this is the inner membrane. This one in between these 2 membrane is the mitochondrial space, and then this is the whole is the surrounded is the matrix, and these are the cristae. These are the infolding of inner membrane. You look at the picture. These are the cristae, shelf-like projection of the inner mitochondrial membrane. This whole is the matrix of the mitochondria. This is again a picture. Outer mitochondrial membrane, this one. This is inner mitochondrial membrane, and then the intermembranous space. This whole is the matrix. These are the shelf-like projection of the inner mitochondrial membrane. It is called as the cristae of the mitochondria. This is the hypothetical diagram to understand you, made easy. That's the mitochondria. Look at the picture. So this is a oval-shaped organelle, double membrane, the outer and inner membrane. This is the intermembranous space, and these are the shelf-like projection of the cristae. That is the inner mitochondrial membrane infolding, and these cristae are attached with the F1, F0 particle. And these F particles, they are concerned for the very important function of ATP synthesis, adenosine triphosphate. And in between you see the matrix having the ribosome as well as the linear DNA chain. Look at the DNA chain, and this DNA chain is the chromosomal material for the self-production of the protein necessary for the mitochondrial function. Now, this is the very beautiful picture of a hypothetical diagram of a mitochondria. Now, this is the 2 model, Beffel model and Crista junction model. It is these models are based on the shape and location of the cristae within the mitochondria. In this Beffel model, you see this is the infolding of the inner mitochondrial membrane having a separate mitochondrial space, inner mitochondrial space. Look at these, it itself, whole of the membrane, inner membrane is folded itself. There is no interconnected part of the membrane, so there is no, look at the picture, Over here, this is a continuous spaces present within the Christie, but in the Christie junction model, look at this tubular formation, or you see a tubule of the infolding of this Christie having attachment with the inner mitochondrial membrane space. Look at this picture. So this junction, this is called a Christie junction. जहाँ पे junction बने हैं वहाँ ये आप देखेंगे it is having no junction, although it is communicate basally toward the intermembranous space. So over here, but over here this is junction. So this mechanism of this system, तो हम इसको mitochondria को मॉडल कहेंगे क्रिस्टा जंक्शन मॉडल. This is a difference. This all together, this membrane is folded to form a shelf-like projection. F1 particles are attached on both models. So ATP, DNA, and ribosomes all are present within these two models. Now we'll discuss the outer mitochondrial membrane. Encloses the entire organelle, is about 60 to 75 angstrom thickness. एंटायर ऑर्गेनाली को बाहर से जो मेम्ब्रेन कवर करेगी उसको हम आउटर माइटोकॉन्ड्रियल मेम्ब्रेन करेंगे. थिकनेस जो है 60 से 75 एंगस्ट्रॉम होगी. इन कंटेन लार्ज नंबर ऑफ इंटीग्रल मेम्ब्रेन प्रोटीन, सो आल्सो कॉल्ड एज पोरीन, विच कम्युनिकेट विद साइटोसोल विद द इंटरमेम्ब्रेनस स्पेस. ये बात आपने याद रखनी है. द आउटर माइटोकॉन्ड्रियल मेम्ब्रेन इज अ पोरस मेम्ब्रेन. इट हैज़ ए वेल नोन communicating channel with the help of integral membrane protein. This is called as porine complex. This porine complex communicates with the cytosol. These porines form channels that allow molecule up to 50 nanometer to pass into the space. तो यही वो बात आ गई कि 50 नैनोमीटर तक जो भी मॉलिक्यूल होंगे वो तो पास कर जाएंगे, लेकिन इससे अबव नहीं पास करेंगे. लेकिन नेचर ने यहाँ भी एक मैकेनिज्म दे दिया. लार्जर प्रोटीन कैन एंटर द माइटोकॉन्ड्रिया टू ट्रांसलोकेटर प्रोटीन ऑन मेम्ब्रेन वाया एक्टिव ट्रांसपोर्ट. ये बात आपने याद रखनी है कि अल्लाह ताला ने फिर जब बड़े मालिक्यूल्स होते हैं उनके लिए ट्रांसलोकेटर प्रोटीन्स हैं. ये भी ट्रांसमेम्ब्रेन प्रोटीन है, लेकिन ये इनका गेट तब खुलेगा जब इनके साथ एक कैरेक्टरिस्टिक सिग्नल पेप्टाइड लगेगा, वो नॉक करेगा और विद द हेल्प ऑफ एक्टिव ट्रांसपोर्ट वो प्रोटीन भी पास हो जाएगी थ्रू एंड थ्रू विद इन द मैट्रिक्स. द आउटर मेम्ब्रेन आल्सो कंटेन एंजाइम मेनली मोनोअमीन ऑक्सीडेज, retinoic insensitive NADH cytochrome C reductase, carnitine hydroxylase and fatty acid CoA ligase. So these enzymes are also located with the outer mitochondrial membrane because they are concerned for the Krebs citric acid cycle, which is needed to synthesize the ATP molecule within the mitochondria. So they will help in transport of the various molecules from the cytosol to inside the mitochondria, and also the fatty acid metabolism, various coenzyme A and many other. So these enzymes are located in the outer mitochondrial membrane. Monoamine oxidase, retinoid insensitive NADH cytochrome C reductase, carnitine hydroxylase and fatty acid coligase. These are the few examples of a enzyme present in the outer mitochondrial membrane. Now look at the picture of this porine complex and translocator protein. Now over here, a larger molecule of a protein that is more than 50 angstrom, जिस वजन होगा वो उसके प्रोटीन जो है इट हैज़ ए करेंगे स्पेशल सिग्नल सीक्वेंस, signal peptide or signal sequence. This special signal where approaches the receptor sites. These are the receptor site. जब receptor site पहुँचेंगे then when attached there will be configuration changes in the translocator protein, which are the transmembrane protein in the outer mitochondrial membrane. What will happen when it gets attached to it? It will open the gate contact sites, and these contact sites are double. That is, the inner mitochondria will also open up by this translocator signal sequence. Where it happen? So this protein, which is larger molecular weight, passes through this translocator protein, transmembrane protein, porine, and with the help of this move inside in the mitochondrial matrix, from where this protein is utilized within the matrix and clip the signal sequence for further use outside. Now over here, then when this will happen over here, this translocation opening up needs energy. ATP molecule is over here to help. When this function is over, what will happen? So when this clip of the outer membrane will again configuration change in the receptor, it will close the gate for no further entry. Whenever entry will occur, this is the signal sequence. This signal sequence has a receptor site for this receptors in the outer mitochondrial membrane. ये इसको recognize करेगा, ये knock करेगा और प्रोटीन ये membrane को बताएगा कि मैं wanted हूँ within the matrix of the mitochondria, मुझे जाने दें. When it recognize it, yes, it is the needed protein for the matrix. But if it happen, it will change this translocator protein for configuration, and the gate will open up. This gate will open up, and protein is move across this through and through toward the matrix to be propagated and used in the matrix. This is the mechanism of a translocator. Over here, ATP again help for the release of energy. Now intermembranous space, अब हम next बात करेंगे. The intermembranous space is space between the outer and the inner membrane. It is also called as mitochondrial space. Ions and sugars in the membrane space is the same as that of cytosol due to free permeability, but H ions are frequently present more. ये बात आज की चूँकि porine, the pore complex it is freely permeable below 50, so cytosolic contents are almost equal in the sugar, and ions are present equally at the cytosol, but the H ions are more prominently present because they are produced constantly within the mitochondrial matrix by the cytochrome chain system. So this cytochrome chain system will produce more H ions. They will be freely moved from the mitochondrial matrix toward the intermembranous. So H ions are more prominently present with this in the intermembranous space. However, large protein must have specific signal and sequence to be transported across the outer membrane, so the protein composition of this space is different from the protein composition of the cytosol. जैसे मैंने पहले बता दिया कि प्रोटीन जो कंटेंट्स हैं उसका डिफरेंशिएशन है between the cytoplasm and that of the mitochondria, because sometimes mitochondria has a specialized protein either produced by its own DNA molecule or from the signal through the nuclear DNA of the cell itself, from where it carries a signal, and this signal is then transported through the translocator protein into the matrix. One specialized enzyme protein named the cytochrome C also located in the space. ये बात याद रखें. The question, one specialized enzyme protein for this cytochrome chain, that is cytochrome C reductase or cytochrome C, it is also localized in this space. Other chain के जो enzymes हैं वो नहीं हैं, लेकिन जो cytochrome C है वो is located within the intermembranous space. Now, this is the diagrammatic presentation. This is the cytosol. This is the inside the matrix of the mitochondria. This is the outer mitochondrial membrane. This is inner mitochondrial membrane. So look at the picture. The more of the H ions are commonly present, and then these H ions are transported across within the ATP molecule into the matrix, from where the ATP molecule is generated. So this is the mechanism, ATP synthase. So this ATP synthase is concerned in F1 particle for production of ATP. We will discuss in the next slide. But if you have to look at a picture, more H ions are present within the intermembranous space. So inner mitochondrial membrane. The inner mitochondrial membrane contains protein with 5 types of function. अब जो inner mitochondrial membrane है उसके सारे के सारे जो है उसके ऊपर protein जो molecules they are attached, they perform this very important function of various 5 function. Oxidation, reduction, reaction जो है, it is concerned for this inner mitochondrial membrane. ATP synthetase, which generate ATP in the matrix in F1 particles, are also located in the inner mitochondrial membrane. Specific transporter proteins that regulate metabolite passage into and out of the matrix. ये भी प्रोटीन जो है वो लोकेटेड होती हैं फॉर द फंक्शन ऑफ transportation of the various molecules from inside to outside. Then fourth is the protein import machinery. Protein import machinery, जो import proteins हैं उनको लाने के लिए भी सारा system जो है it is present within the inner mitochondrial membrane. Mitochondrial fusion and fission protein. माइटोकॉन्ड्रिया जो फ्यूज होते हैं या उनको ब्रेक डाउन है, उनकी जो प्रोटीन्स हैं जब माइटोकॉन्ड्रियल लाइसिस होती है या bigger molecule of the mitochondria and fusion and fission, their concerned proteins are also located in the inner mitochondrial membrane. Unlike the outer membrane, the inner mitochondrial does not contain porine. It is highly impermeable to the all molecules. ये बात याद रखनी है कि inner mitochondrial membrane is highly impermeable. It is only and only give way to the translocator protein जो कि सिग्नल पेप्टाइड लेके आएंगी, अदरवाइज नो एंट्री. इट इज स्ट्रिक्ट रूल दैट नो एंट्री बिकॉज अंदर मैट्रिक्स के अंदर जो है वेरी वेरी इंपॉर्टेंट फंक्शन इज गोइंग टू बी परफॉर्म. वहाँ पे कोई मुदाखलत नहीं होगी सिवाय जो वांटेड है. तो वांटेड जो है वो थ्रू ट्रांसलोकेटेड प्रोटीन, लेकिन जो पोरीन्स हैं वो सिर्फ आउटर मेम्ब्रेन पे होगी, इनर पे नहीं होगी, ताकि कोई भी फ्री चीज़ आ जा ना सके. Almost all ions and molecules require special membrane transporters to enter the exit matrix. तो ये बात मैंने पहले बता दी. तो जब चीज़ निकलनी है, आयन्स भी इवन या प्रोटीन्स भी या कोई भी मॉलिक्यूल, उसके लिए स्पेशलाइज्ड ट्रांसपोर्टर प्रोटीन्स बनी हुई हैं जो कि उसको एग्जिट एंट्री देती हैं. सो इनर इज मोर स्पेसिफिक मेम्ब्रेन, only give way to these specific molecules. Now, what are the cristae? Cristae are the chef-like projection in the matrix to increase the surface area. इनका फंक्शन क्या होगा क्रिस्टी का? जो प्लाज्मा जो मेम्ब्रेन है इनर माइटोकॉन्ड्रियल मेम्ब्रेन, उसकी जो इनफोल्डिंग है वो इंक्रीज करेगा सरफेस एरिया, ताकि जो F1 particles are more numerous present over there, so increase the surface area, increase activity. Now, these cristae are the site for the F1 particle activity of ATP molecule, like muscle cell. It is more prominent present in the muscle cell cristae, more F1 particle. Attached to the cristae are the F1 particle oxysomes. So oxysomes are other name of the F1 particle. So these are the site for the ATP production. ATP synthase is the enzyme which convert the ATP molecule to ATP molecule. F1 particle the site for ATPase activity. F1 particles have a head and stalk. ये आपने सबने पता है कि head and stalk. The stalk has a tunnel protein meant for HN influx. तो F1 particle में जो head है, it is location for the ATPase, that is the adenosine triphosphatase, which is enzyme located, whereas the stalk is concerned for the influx of HN through tunnel protein complex. ये हम बाद में दिखाएंगे आपको. Electron transport chain and F1 particle shape and its location. Look at the picture of this inner mitochondrial membrane. Inner mitochondrial membrane is also a double membrane of lipid protein. So look at this is the double layer of phospholipids. The tails are toward facing each other, where head are toward the space or in the matrix. So this is typical plasma membrane, but it is the inner mitochondrial membrane. Now these all are the transmembrane and peripheral proteins which are concerned for the electron transport chain protein. Look at the picture. So this is the electron transport chain protein 1, 2, 3 and 4, and one protein and Q enzyme Q, and then the cytochrome C. This is the one protein which is in the intermembranous space. Look at the picture. This is the transmembrane protein. This is the peripheral protein. Q enzyme is also peripheral protein. 1, 2, 3 are the transmembrane protein, and cytochrome C is the only protein of the electron transport which is located within the intermembranous space. Now, this is the F1 particle, this is the stalk, and this is the head. So this head is typically for concern for the ATPase activity. So ADP molecule with PO4 addition, they will added up to form the ATP molecule. So over here the H chains are required. So this H chains are produced by the electron transport from the help of NADH, nicotine adenine dinucleotide hydrogen. It will give hydrogen molecule and become positively charged. That is the NAD, from where H chains are released from this H chain, then activates the second coenzyme and second protein, flavine adenine dinucleotide hydrogen. It will convert into again releasing the H chains. So H chains are freely present within the mitochondrial matrix, from where it is transported out within the intermembranous space again. So these are the translocator protein which carries the H chain produced by the FADH2 and NADH. So they will release the H chain from where it is transported out, because over here it is the active ingredient, energy is needed over here, from where pours into the toward the H chain, from where more concentration of H chain. So this H chains are very, very urgently needed for the F1 particle to be transported within the again in the matrix. After passing, it will change in the configuration of the F1 particle head. When this head is changed, the active side will be placed, and ADP molecule that is present within the matrix with the help of phosphate convert into ATP molecule, and this ATP molecule is released to be used within the cytoplasm. So this is the conjunctional activity. ये दोनों एक्टिविटीज साथ साथ चल रही हैं. एच चैन प्रोडक्शन भी हो रही है, इलेक्ट्रॉन ट्रांसपोर्ट चेन, उससे एच चैन प्रोड्यूस भी हो रहे हैं, रिलीज हो रहे हैं विद इन द मेम्ब्रेनस स्पेस, मूव होके टुवर्ड द स्टॉक जो है वो जा रहे हैं. स्टॉक के अंदर टनल बनी हुई है, वहाँ से टनल से वो एच चैन जो है मूव करते हैं. वो मूव करने से वो रोटर बना हुआ घूमता रहता है. वो घूमने से साइड्स जो है एक्टिव हो जाती हैं हेड के ऊपर. वो साइड एक्टिव जो है वहाँ एडीपी मॉलिक्यूल आके जुड़ता है विद द हेल्प ऑफ addition of phosphate 1 molecule. So when it occurs, then the ADP is converted into adenosine triphosphate, which is the active ingredient of the energy dependent molecule. So ये बात याद रखनी है. Most of the activity is within the mitochondrial matrix because NADH, FADH present. This from where they break down with the various enzymes. So this H chains are produced and translocated within the membranous space. These are the cytochrome 2 coenzyme. So they will transport this toward the cytochrome C reductase, from where again the electron is produced, and this electron H chain and H and water plus oxygen will produce the water. So 2 free hydrogen ion, 2 electron exiting electron transport chain, and half of the oxygen molecule will join to form the water molecule. So excess of electron they are needed to form the hydrogen ion. So will the hydrogen ion will combine with the oxygen to form water. This process, this reaction is also taking place within the matrix. Again, this hydrogen is again utilized in the membranous space to move inside this tunnel toward the F1 particle, from where it is formation takes place in the ATP molecule. Now this is the mechanism of the transportation. Inshallah we will discuss next time in the next lecture. Thank you very much.